Casual conversational cartoon curriculum, very concise and relatively correct, as simple as possible, as soon as possible, anatomy and physiology. Here's our fourth little installment on this thing right here. It's very short because I forgot to mention this and this right here about the cell membrane. There are structures that the cell membrane can create, structures such as microvilli, cilia, and flagella. Microvilli are finger-like projections of the cell membrane that increase the surface area of the cell dramatically. You find this in places of the body where you want to absorb things, such as the inner lining of the small intestine and also, say, inside the kidney where you're absorbing materials from the filtrate, the almost urine material. Other like structures may be thread-like, and these thread-like ones, hair-like ones, are called cilia. Now, these things are for moving material past the surface of the cell. So not for absorption, for moving materials past the surface of the cell. This is very obvious in the fallopian tubes. It moved the zygote, then the morula, then the blastula, etc., down the fallopian tube towards the uterus. Also in the back of the nasal cavity, also inside or lining your trachea, sweeping, in the case of the respiratory system, mucus towards your throat. And then a simple example right here of a flagellum would be on a sperm cell. Again, a single thread-like structure that while twirling acts like a propeller. Well, three other features right here of the cell membrane that are unique. When two cells are parked next to each other, sometimes they connect. And when they connect, it could be called a tight junction. Now, a tight junction is like a ziplock. It's like a tongue and groove. It's where one thing protrudes out, the other thing protrudes back, and you form a seal. Now, tight junctions tend to be found in places where you don't want materials to move between the cells. So at surfaces of the body, like the inner lining of the stomach, the inner lining of the small intestine. You'd say that if these are like two houses right here, and here's a street, and here's your backyard, this thing right here is like the fence that prevents things from moving between the houses through the alley going to your backyard. So again, tight junctions right there. Again, they act like ziplocks. Another type of junction is called a desmosome. A desmosome is like a button or rivet. It isn't a line that's creating a continuous fence barrier. It's just a spot. And in this spot right here, you just have some protein that can be anchored to a protein of another cell. Now, this is like a rivet or a button in that you're connecting two objects not continuously along a line, but at these spots right here. You tend to find desmosomes in places where cells are being pulled apart from each other. So in cardiac muscle tissue, there's a lot of desmosomes where one cardiac muscle cell is hanging on to another cardiac muscle cell. And the last example right here of the junction would be a gap junction. The gap junction is actually a channel between the cytoplasms of two neighboring cells. Now, embryologically, there were a lot of gap junctions, say in the morula stage, because the individual cells weren't capable of taking care of themselves. They had to share things. Gap junctions still exist inside the adult body. The most notable example, again, is cardiac muscle tissue, where one cell is connected to the cytoplasm of another cell, thus allowing a depolarization, an impulse on one cell to sweep to the neighboring cell, thus making it unnecessary to have nerve fibers going to each individual cardiac muscle cell.